Three, two, one, go.
Sure. Good day, everyone, and welcome to this time lockdown digital school. My name is Katla Matibe, and I'll be presenting the grade 10 life sciences lesson to you guys. Welcome back to those who have been with us, and let us welcome our new members. If you are new to this platform or to this class, welcome and thanks for joining us. And those who have been with us, thanks for remaining loyal and for coming to class early and for participating also. Thanks, guys. So, guys, do remember to use my email address that is at ls.wisnet@gmail.com. Use that email address for anything related to life sciences. So if you need help or these slides, just hit me up on that email address and I will gladly help you guys. All right. Now, as you know that on Monday, we started a new topic, which is on cells under the strand life at the molecular, cellular, and tissue level. We are still on that topic and we are continuing. So we are continuing with cell structure and function. And today we are going to talk about the cell membrane, fluid mosaic model, and movements across membranes. So that is our topic for today. So it will be based on cell membrane, fluid mosaic model, and movements across membranes. So at the end of this lesson, what I want you guys to do is to be able to define concepts that are highlighted in bold black. So those of you who have been in this class ever since we started the life sciences lessons, define concepts. So as you know, in life sciences, terminology is important. In every test, you have to define a term, whether you like it or not. So guys, be able to define concepts that are highlighted in bold black. I'm sorry if I'm breaking up. It is most probably my network, my connectivity, guys. Forgive me for that. But there is nothing I can do. Just try to listen carefully or closely there. So as I was saying, terminology is important. You guys should, I encourage you guys to have a terminology book and write all the terminology or the terms for each and every section that we cover. So now we are on term 2 stuff. You can have a book and have a section on cells and write terminologies under that section. You will never go wrong, trust me. So I want you guys to be able to define concepts as the liquid part of the cell, to differentiate between protoplasm, nucleoplasm, pardon me for that, and cytoplasm. And I want you guys to recognize the difference between a cell wall and a cell membrane. Remember yesterday we touched base on the cell wall. We talked about the cell wall. So you should be able to recognize the difference between a cell wall and a cell membrane that is in structure, function, and the location. And I want you guys to understand the structural component of the fluid mosaic model that different substances will move in different ways to differentiate between diffusion, osmosis, and active transport, and to understand the process for each movement. So that is what I want you guys to be able to do at the end of this lesson. Those are our objectives. Now, just to quickly recap, yesterday we talked about the cell wall and we finished off our microscopy calculations that was based on magnification, the field of view. I hope you guys still remember that. That is for your practical. So on cell walls, we said that all cells have the same basic structure. So they are all surrounded by a cell membrane and they contain the cytoplasm and other cell organelles. So cells have different sizes, shapes, and structures in order to carry out specialized functions. And we said the cell wall is only found in plant cells and it is made up of cellulose. You know that cellulose is a type of carbohydrate, that is a type of a polysaccharide. And the cell wall
Teacher, please check your audio. You have gone completely silent.
Yeah, it was network. Guys, I'm sorry, I'm back. Sorry about that. My connectivity. I'm back. Can everybody hear me now?
Yes, we can hear you again.
All right, thank you. I'm sorry, guys. It's really bad. I don't know what I can do now. Forgive me for that. So as I was saying, know the structure, function, and the location of the cell wall. Know that. So I gave you a homework and I said you guys should tabulate 5 differences between plant and animal cells. Let me just quickly go back. Could you quickly give me the 5 differences between plants and animal cells? The differences between plants and animal cells? What are those? What are the differences between plants and animal cells? Lucasesi says a plant has cell membrane and animals don't have. That is not quite correct. Zach says plants have cell walls, animals do not have cell walls. Yes, so Lucasesi, plants have cell walls and animals do not. So plants and animals have both, they both have the cell membrane. The only difference is the cell wall. So the cell wall and the cell membrane are not the same. They are not the same thing. Okay. So plants have a cell wall and animals do not have a cell wall. Their similarities, they both have the cell membrane. It's all right. So what are other differences between plants and animals? First one we name the cell wall. What are the other ones? Yes, Zach, I'm waiting and time is not on our side. I am going to move to just quickly give you guys the corrections, but because you guys seem to not have the answers. So Gundo says plant cells have more of a fixed shape. So they have a regular shape as compared to animal cells. Animal cells do not have a regular shape, but rather an irregular shape because they do not have a cell wall. So Charles says, and human cells. Remember human cells, humans are animals. So plants, yes, cells can be denoted as jail. But then human cells are the same as animal cells. They form a part of animal cells. So Letao says the plant cell has a regular shape and animal cells have, I think you meant to say, an irregular shape. So Zach says animal cells have more mitochondria as opposed to plant cells. Lusa says plants have one large vacuole and animals have small vacuoles. So a numerous number of that. Gunda says all animal cells don't have chloroplasts. So yes, plant cells have chloroplasts, but animal cells do not. I'm sorry, Charles, it's connectivity. All right, so guys, thanks for the responses. So these are your solutions. Plant cells. Under plant cells, we know that a cell wall is present, or the outermost layer is the cell wall. It has a regular shape. It has a single large vacuole. A chloroplast is present, and plastids are present also. So we talked about plastids yesterday. Your leucoplasts, your chromoplasts, those are present in the plant cell. So in animal cells, no cell wall is present, or the outermost layer is the cell membrane. It has an irregular shape. It has small vacuoles. No chloroplast is present, and plastids are absent. So it does not have plastids. Now moving on to today's work. We are continuing with cell structure and function. And we're going to talk about the cytoplasm. So as we mentioned yesterday, that the protoplasm is the colorless material comprising of so Charles is asking, what do I mean by shape? The shape of a cell. I mean the structure. Is it regular? Is it a rectangular or circular or a square? That is what I mean by shape. So for plant cells, they have a regular shape. It means that you can tell that this is a triangle or a square or any other shape that you know of. But then for animal cells, you cannot tell what shape it is. So it is a shape you cannot name. It's just irregular. So that is what I mean by shape. So for regular, you can tell that this is a square, rectangle, triangle, or circle, any other shape type that you know. And for irregular, you don't know what that shape is. Yes, Tyree, sometimes it's not counted as a shape for animal cells. So let us continue. The protoplasm is the colorless material comprising of all the living parts of the cell. and it is made up of the nucleus, cytoplasm, and other organelles. So as you guys see, the term is in bold black, so take note of that. Everything within the nucleus that is not part of the nucleolus is called the nucleoplasm. So this is the substance of the cell nucleus. So everything within the cell that is not part of the nucleolus. We will talk about the nucleus tomorrow. So you get a more better understanding of what I mean by the nucleus, the nucleolus, and the nucleoplasm. So the affix plasm, it refers to a substance or a living substance of a cell. So as you can see that we're talking about the protoplasm, and it is a colorless material, so a colorless substance comprising of all the living parts of the cell. The nucleoplasm, it is the substance of the cell nucleus. And now the cytoplasm, which is the fluid part of the cell that is made up of almost 90% of water and dissolved organic and inorganic compounds. So the term or affix plasm, it refers to a substance or a living substance of a cell. So it can be either a prefix or a suffix. All right, so as a prefix, we know the terms plasmodesmata. We talked about that yesterday. And as a suffix, it is your protoplasm, your cytoplasms. All right. So the cytoplasm is the fluid part of the cell, which is made up of almost 90% of water and dissolved organic and inorganic compounds. So it may occur in two states. So the cytoplasm may occur in two states. Firstly, the sol, which is a more fluid or liquid state. And secondly, the gel. So it's a more jelly-like viscous medium. A more jelly-like or viscous medium. Charles is asking, do you get sizes that are in layman's terms plus size? Yeah, you do, because they differ in sizes and shapes. So they differ in sizes and shapes. Do not know what plus size is. So larger cells, we do have larger cells, so they differ in sizes and shapes. So the main function of the cytoplasm is to hold together the organelles which make up the cytoplasm. So it holds up together the organelles which make up the cytoplasm. And it also nourishes the cell by supplying it with salts and sugars and provides a medium for metabolic reactions to occur. So it also nourishes the cell by supplying it with salts and sugars, and it provides a medium for metabolic reactions to occur. All right, so looking at the functions of the cytoplasm. So Charles is asking if metabolic reactions, can they be measured as a rate of time? Yeah, it is a smart question, and they can. And actually they are. So when we mean the rate of reaction, we actually mean the rate of the time. So it is the rate of time when we talk about the rate of reaction. So now he's asking if it can be calculated in derivatives. I do not know that. You can go check it out for us or search it for us and come back, give us the answer to that. So the functions of the cytoplasm provides mechanical support to the cell by exerting pressure against the cell's membrane, which helps keep the shape of the cell. So this pressure is known as the Turger pressure. So the Turger pressure is the pressure against the cell membrane, which helps keep the shape of the cell. The other function is that it is the site of most cellular activities, including metabolism, cell division, and protein synthesis. So it also contains ribosomes, which assist in the synthesis of proteins, and it acts as a storage area for small carbohydrate, lipid, and protein molecules. And lastly, it suspends and can transport organelles around the cell. So this is a diagram of the cell. This is our cell membrane. And we can see the protoplasm. Remember we said the protoplasm contains, or it is made up of the nucleus and the cytoplasm and other cell organelles. The nucleus therefore has the nucleoplasm, which is the substance of the cell nucleus and the nucleosol, and the cytoplasm made up of the cytosol. All right, so this is just a diagram to show you guys of what we have been talking about. So in this case, this is an animal cell because there is no cell wall. So this is how an animal cell looks like. We cannot tell what shape this is. This is an irregular shape, and hence the outermost layer is the cell membrane. The outermost layer is the cell membrane. So Megan is asking, can a structure have a double membrane? Yes, it can. Remember with the plant cell, we said that it has the primary cell wall and the secondary cell wall. So that is a double membrane. So Charles is asking, can we separate the nucleus from the cell membrane? Yeah, we can. We can, but that is deep microbiology. And so the cell membrane. So the cell membrane is also called the plasma membrane. Take note of this. It is also called the plasma membrane or the plasma lemma, if you come across that name in your textbooks. It can be called the plasma membrane or the plasma lemma. It physically separates the intracellular space, which is the inside of the cell, from the extracellular environment, which is the outside of the cell. So all plants and animals have cell membranes. Lucasesi, you can see that. All plants and animal cells have cell membranes. So the cell membrane surrounds and protects the cytoplasm, like we saw in the diagram, which was on the previous slide. So all the exchanges between the cell and its environment have to pass through the cell membrane, meaning that it is permeable. So it is a semi-permeable membrane and controls the movement of substances into and out of the cell. It controls the movement of substances into and out of the cell. It is a semi-permeable membrane. What does that mean? Semi-permeable. What does it mean? What does semi-permeable membrane mean? So Charles is asking, with all of this research that we're doing on cells, how come we are unable to clone people? I don't know why. Because human beings are, or human cells are still hard to clone. I don't know. Touch that. Miss P, we are doing cells, and we are talking about cell membrane. So the cell structure and functions of cell organelles. We are talking about cell membrane. So Mila says it allows certain substances to pass through. So semi-permeable membrane means that it allows a certain types of substances to pass through. Not all can pass through. So the cell membrane is strong and flexible to provide mechanical support, and it allows cells to grow and move respectively. So the cell membrane is strong to provide mechanical support, and it is also flexible to allow cells to grow and move. Now, maybe Charles can be the first to clone people. Charles, I advise you to do a research on that and be the first to clone people. That will be amazing. So this is all on cell membranes. It is a semi-permeable membrane. So remember I said you guys should be able to differentiate between the cell wall and the cell membrane. So the cell membrane is a semi-permeable membrane. It separates the inside of the cell from the outside. So Megan is asking, can we specialize cells? Can we form specialized cells? Go deeper into that question. Do not understand what you're trying to say. Specialized cells like what? Now moving on, the fluid mosaic model. So this model represents the structure of the cell membrane. So the structure of the cell membrane can be represented by the fluid mosaic model. And this model was proposed by S.J. Singer and G.L. Nicholson. Scientists cannot see the So Sophie's asking, what about cancer cells? Can't you eliminate the amount of cancer cells? We can't. We cannot. That's why there is chemotherapy. And if you have a tumor, they remove that tumor to reduce the number or the amount of cancer cells in the body. So we can eliminate those by chemotherapy, but not all of them. So it suppresses the function of these cancer cells. Yes, meristematic cells can differentiate to form specialized cells. So we're going to talk about types of cells later on. So I see where you're going, Megan. I see where you are heading at. So our bodies can form specialized cells, all right? And in the lab also we can. So the cell membrane is composed of a double layer. We call the double layer a bilayer because bi, the prefix bi is for double. Where's my cursor? Okay. So the double layer is called the bilayer of special lipids or fats. They are called the phospholipids. So phospholipids consist of a hydrophilic or a water-loving head and a hydrophobic, which is water-fearing tail. It consists of a hydrophilic head and a hydrophobic tail. So a water-loving head and a water-fearing tail. So the hydrophobic head I meant to say the hydrophilic. This is supposed to be the hydrophilic. That is supposed to be the hydrophilic. Right, so the guys, I'm having difficulty maneuvering around my slides here. Bear with me for that. I don't know what's happening. I don't know what's happening. Do not know what is happening. So the hydrophilic head of the phospholipid is polar, meaning that it is charged and can therefore dissolve in water. And the hydrophobic tail is nonpolar, meaning that it is charged, it is not charged and cannot dissolve in water. So by hydrophilic, we mean that it can dissolve in water, hence it is water loving. And by hydrophobic, it cannot dissolve in water. So the lipid bilayer forms spontaneously due to the properties of the phospholipid molecules. So the lipid bilayer, so that densely due to properties of the phospholipid molecules. It forms spontaneously due to properties of the phospholipid molecules. So in an aqueous environment, the polar heads try to form hydrogen bonds with the water. Remember, hydrophilic heads. So by polar, we mean that they are charged and are water loving. So they try to form hydrogen bonds with the water, while the non-polar tails try to escape from the water. I'm sorry, Chloe, I'll repeat that. So the lipid bilayer forms spontaneously due to the properties of the phospholipid molecules. So what are the properties? The properties are that the phospholipid molecules consist of a hydrophilic head and a hydrophobic tail. So the hydrophilic head is the And the hydrophobic tail is the nonpolar tail, meaning that it cannot dissolve in water. So remember, the phospholipids are special lipids or special fats, and fat cannot dissolve in water. So the hydrophobic tail is nonpolar, and it tries to escape from the water. Right. So the problem is solved by the formation of a bilayer, because the hydrophilic heads can point outwards and form hydrogen bonds with water. And the hydrophobic tails point towards one another and are protected from the water molecules. So you guys might be wondering, what am I talking about? Let me show you this diagram here. So this is the diagram of what we're talking about. These are the hydrophilic heads. They interact with water, so they point towards each other from the water. These are exposed to the outside or external surface of the membrane, and the hydrophobic tails are inside, away from the surface. So this is Charles, you are confusing the class with that question. So this is the structure of the fluid mosaic model, which denotes the structure of the cell membrane. It denotes the structure of the cell membrane. So the fluid mosaic model denotes the structure of the cell membrane. Can explain it using this model. So this is our fluid mosaic model. The polar heads, the non-polar tails are inside. These ones, they do not react with water, and these ones on the I'm sorry if I am breaking up. Yo. If I am still breaking up, it is because of my network. So I'm sorry about that, guys. It's my network. Okay.
Teacher, please just check your audio. We cannot hear you at the moment.
Guys, I'm sorry about that. My network is so bad. It is really bad. Yes, Zach, everybody's using the internet for everything now. So it's quite busy. And I'm sorry about that, but I'm back. Sorry about that. So we said that the cell membrane performs several functions for the cell, and some of those are that it controls the movement of substances into and out of the cell. It provides the cell with mechanical support, and it is flexible, meaning that it allows cells to grow and move. Now, to talk about this movement across membranes. So for the cell to carry out its function, substances tend to enter and leave the cell. So movement of substances across cell membranes is necessary as it allows cells to acquire oxygen and nutrients, to excrete waste products, and control the concentration of required substances in the cell. For example, oxygen, water, hormones, your ions, etc. So for the movement of these substances to the membranes of the cell, which are semi-permeable, and the key processes through which such movement occurs include diffusion, osmosis, facilitated diffusion, and active transport. So we're going to delve deeper into those. So diffusion of substances from a region of high to a region of low, said to occur down a concentration gradient. So when we mean down, or maybe your textbook may say it occurs along a concentration gradient, we mean that it moves from a region of high concentration to low concentration. So that is what we mean by along or down a concentration gradient. So fusion is a passive process. You should know what we mean by passive process, which means that it does not require any energy input. Yo, sorry about that again. So guys, it does not require any energy input. I feel like we have 3 minutes remaining, so I will just end on this slide because network is just not on my side today. So it can occur across a living or non-living membrane and can occur in a liquid or gas medium. So diffusion can occur in a liquid or gas medium. So due to the fact that out of the cell. So examples of substances moved by diffusion include carbon dioxide, oxygen, water, and other small molecules that are able to dissolve within the lipid bilayer. So guys, give me an example of diffusion. An example of diffusion that occurs in our daily lives. air freshener. So Zach says the air freshener. So use the air freshener, it moves from a region of high concentration to a region of low concentration. So it evenly spreads around the room. So if you're spraying in a room, from where you're standing, there's a region of high concentration and it spreads around until it is evenly spread. So it spreads to lower concentrations. Oxygen and carbon dioxide in our lungs. Thanks for that. That is a nice example. Juice again. So juice is a liquid. So when you pour your orals, which is concentrated in water, you mix it and then it is evenly spread. Thanks guys for guys, because of time, I will end here because network was not on my side. We'll continue with other movement across membranes tomorrow. And hopefully finish it off. I'm sorry about what happened today. Have a nice afternoon and see you tomorrow, same place, same time. Thanks for bearing with me, guys. You are awesome. Goodbye.